Hello biology students, and welcome to video 2 of the cell unit. So in this video we're going to be focusing on a couple main guiding questions. We're going to say, okay, what are the membrane-bound organelles that are found in the eukaryotic cell? And then we're going to be looking at what are the functions of these organelles and what relationship exists between the form or the structure and their function. So let's get going. So here is a typical eukaryotic cell. So you can see there's lots of different organelles that we have to look at and explore the functions of. So the first one, the biggie, the one that defines a eukaryotic cell, is the nucleus. Okay? So this is found in all eukaryotic cells, so plants and animals. And primarily it acts to store and protect DNA. So when DNA is in the nucleus, it's in the form that we call chromatin, which is basically long strings of DNA. And because the nucleus has DNA, and DNA gives us all our genes and all our traits, really the nucleus controls most of the functions of the cell. So its structure, what it looks like, it has a double membrane, which means there are two membranes around the nucleus. And we call this double membrane the nuclear envelope. And in the nuclear envelope there are pores or holes, and these pores let materials get in and out of the nucleus. Here's what a nucleus looks like. So you can see there are two membranes. It's a double membrane. So you can see there are two layers that are really close together right here. And then in this double membrane, there are these pores, so things can get in and out. If we look, we see DNA is in this stringy form, all these strings in the nucleus. That's the DNA, and we call it that chromatin. Yes, and that's the nucleus. So inside the nucleus, there's this little section that we call the nucleolus. So you can see the nucleolus is this dark purple section inside the nucleus. This is found also in all eukaryotic cells, and this is where the information is stored to help make ribosomes. Okay, so this is a specific area of the nucleus that stores the information to make ribosomes. So its structure is a dense, really condensed area inside the nucleus. You can see that the nucleolus is this dense section in the center of the nucleus, and here is a pretty cool so that's electron microscope photo of the nucleus, and you can see here it's the nucleolus. Okay. So the next organelle is the mitochondria. This is also found in all eukaryotic cells, and its main function is the site of cellular respiration. So cellular respiration is the process by which we make energy. So this is where we're going to break down all that glucose that we eat and use it to make ATP, which is the energy currency of the cell. It also has a double membrane, just like the nucleus. And active cells, so things like muscles that need lots of energy, have lots and lots of mitochondria. This is what the mitochondria looks like. You can see it has this one kind of flat membrane on the outside, and then it has this weird folded membrane on the inside. So it has two membranes, just like the nucleus, but one is all folded up. Okay. So the next organelle are cilia. They're found in some prokaryotes and some eukaryotes, but not all prokaryotes, not all eukaryotes. And what they do, they're these little hairs that move materials alongside the outside of the cells, okay? And they also can help to move the cell itself a little bit. So one place where you find these are in your windpipe, in your trachea, in your throat. So you can see if I have all the little cells that line your windpipe, so here's where the air would go in and out, right? And here's the cells that are lining the outside. So what they have all these little hairs sticking out called cilia. And what these do, that if you get dust in your throat, these little hairs beat together, and they help to beat the dust back up out of your throat. So there are numerous short hairs that are sticking on the surface of the cell. So flagella is very similar to cilia. So what a flagella looks like, if I have a cell here, a flagella is one long kind of tube-like structure that kind of looks like a hair. It's found on some prokaryotes and some eukaryotes. And the function is just to move the cell. So you can kind of see this little tail thing would whip back and forth and can move the cell through some liquid. You can see here at the cilia, these little hairs on the outside of the cells that help to move things on the outside of the cell. And here's a flagella on the end of a sperm cell that helps the sperm cell to move through liquid. The next organelle is the cytoskeleton. It's also found in all eukaryotic cells. And if we break it down, we see that there's two roots here. Cyto, which means cell, and skeleton, which means skeleton. So it's the skeleton of the cell. Basically, this gives shape to the cell, okay? So it helps to support the cell. It allows cells to move. It's the anchor or the tracks on which some organelles move, and it provides rigidity and reinforcement. Okay, and what the structure is basically a network of protein fibers that give the cell structure and they act as tracks for things to move in the cell. So there are three main components that make up the cytoskeleton. There are little tiny fibers. These are called microfilaments. There's medium-sized fibers called intermediate filaments, and large fibers called microtubules. Okay, so the next set of organelles that we're going to talk about are part of what's called the endomembrane system. This is a collection of organelles. There are many organelles that make up the endomembrane system. And basically, this is the system, all the organelles that are going to be working together to manufacture and distribute proteins. And as the proteins are being made, they're going to move from one organelle to the next via what are called vesicles. The vesicles are going to be little sacs that move the protein from one organelle to the next as it's being made. This endomembrane system divides the cell into different compartments, and different parts of the protein-making process occur in different sections of the cell. And again, vesicles we're just going to define as membranous or sacs of membrane, basically. So let's look at this. Okay, so the first organelle in the endomembrane system is the smooth ER, which stands for smooth endoplasmic reticulum. Let me write that down. The smooth ER is found in all eukaryotic cells, and it has tons of functions, okay? The smooth ER is where lipids and hormones are synthesized, so where they're made. So things like the ovaries and the testes, which make estrogen and testosterone, have tons of smooth ER in those cells because they have to make lots of hormones. In some cells, the smooth ER helps to detoxify drugs, so the liver has tons of smooth ER. And the smooth ER can also store calcium ions. Okay, it's located next to the nucleus in the rough ER, and it's smooth and folded. So if we look at a picture, so the smooth ER is over here. So it's called smooth because there are no bumps on it. Notice that this whole section here, that's the rough ER. It has ribosomes on it, which look like bumps, so that's the rough ER. But the smooth ER is this section over here that's bumpless. So the rough ER, or rough endoplasmic reticulum, is also found in all eukaryotic cells. And this is going to function to produce in membrane production. So we're going to make a new membrane. It modifies and packages proteins. So remember, the ribosome makes the proteins. Okay? Then the ribosome sends the proteins to the rough ER, where they're packaged and modified. It's located right next to the nucleus, and it's covered in ribosomes, which kind of look like bumps, which is why it's called the rough ER. Okay, so if you kind of start to look at the protein production process, the ribosome are here, so the ribosome, okay? The ribosome is going to make the protein, and because the ribosome is sitting right on the surface of the rough ER, as it makes the protein, it's going to dump it right into the inside of the rough ER, where the rough ER is going to fold the protein and then modify it. When I say modify it, I mean it's going to add sugars to the surface. You can see I've added these little green sugars. And then the rough ER just pinches off and becomes a vesicle. So this is a vesicle, and now the protein will go to the next organelle in the chain, which is going to be the Golgi. So we can just see the same thing here. So this is the rough ER. We can see these dots are the ribosomes. You can imagine that they're going to make proteins. The proteins are going to go inside the rough ER, get folded and modified, and then eventually the rough ER will pinch off into a vesicle to send the protein on its way. So the vesicle then will drop the protein off at the Golgi. So the Golgi apparatus is found in all eukaryotic cells, and you can kind of think of it as the cellular post office. It's going to receive that protein from the ER, and it's going to add a chemical address. So basically, it's going to add a little sugar molecule to it, and that sugar molecule will tell the protein where to go. So there might be one chemical address that's added if it's supposed to go to the nucleus, and one chemical address added if it's supposed to leave the cell. Okay? So once it has its address, it's put into another vesicle, and it's shipped wherever it's supposed to go. So its structure, the Golgi apparatus kind of looks like stacks of pita bread. So if we look at a picture, you can see these kind of look like pita pockets all stacked up on top of one another. So a vesicle is received from the ER. The protein is dumped into the Golgi. a chemical address is added, and then it's put into another vesicle and sent on its merry way. Here's another picture showing you the same thing. So then finally, sometimes when we make proteins, the protein's job is to be an enzyme that digests food particles or digests things. So if I have a vesicle that contains enzymes that break stuff down, that vesicle is called a lysosome. This is found in all eukaryotic cells and mostly in animals. There are some in plants, but they're mostly in animals. And what they contain hydrolytic. So hydro meaning water, lytic meaning to break. So it's gonna have enzymes that do hydrolysis. Okay? And this is the place things are gonna go if we want to break stuff down. So, these enzymes are going to digest food particles, destroy bacteria, recycle damaged shells. They can function in embryonic development. For example, when you're developing, you originally have webbed feet, and lysosomes can be used to break down all those webs between your fingers. Okay, so these things look like circles. So if we look at a picture of one, here's one here, and you can see that they contain enzymes that are going to digest things and break them down. Okay. And finally, we can also have vesicles that are large, and these we call vacuoles. So they're found in all eukaryotic cells. In plants have one giant vacuole, and animals have many, many small vacuoles. And these are kind of like storage tanks. So you can store food in them, we can store waste, and the plant ones can also pump out excess water. And they're basically sacs that are a little bit bigger than vesicles. Okay. So now that we've talked about the endomembrane system, I just want to summarize it a little bit, because the protein production process can be a little bit confusing. If I'm going to make a protein, there are several things that have to happen. First, the part of the DNA that codes for protein has to be copied. So I'm going to copy my DNA message into RNA. However, that RNA is stuck in the nucleus. So the next thing the RNA has to do is it has to leave the nucleus, and it does that by going through a nuclear pore in the nuclear membrane. And then the RNA finds a ribosome so I can build my protein. Once the protein's built, it will be folded and modified in the rough endoplasmic reticulum. Okay, and then once it's folded and modified, it will be put into a vesicle and sent to the Golgi. The Golgi will add the chemical address and ship it out in a new vesicle. If that vesicle contains a protein that's a hydrolytic enzyme, it will stay the vesicle will stay in the cytoplasm, and it will actually be a lysosome. But if it contains any other protein, it will just go wherever it's supposed to go. So it might, leave the cell, it might go to another part of the cell, etc., etc. So one more time, let's look at that process. So the end part of that process now is after the protein is made in the ribosome, it will be folded in the rough ER, sent to the Golgi. If that protein's a hydrolytic enzyme, it will stay in the cell as a lysosome, and if not, it will leave the cell. So that's it for video 2. I'll see you in video 3.